Next topic, mismatched repair. DNA mismatch repair or MMR is a system for recognizing and repairing erroneous insertion, deletion, and misincorporation of bases that can arise during DNA replication and recombination, as well as repairing some forms of DNA damage. Mismatched repair is strand specific. During DNA synthesis, a newly synthesized daughter strand will commonly include errors. In order to begin repair, the mismatch repair machinery distinguishes the newly synthesized strand from the template parental. Examples of mismatch bases include a GT or AC pairing. Mismatches are commonly due to tautomerization of bases during DNA replication. A DNA mismatch generates due to the incorporation of an incorrect base during the DNA replication process. The mismatch repair system uses the methylation on the parental strand as a signal to leave that strand alone and to correct the nearby mismatch in the unmethylated progeny strand. MUT-A alpha recognizes base-base mismatches, and MUT-L alpha nicks the 3' or 5' side of the mismatched base. The nicked DNA segment is then excised by the XO1 exonuclease. In cooperation with the single-stranded DNA binding protein RPA, DNA polymerase delta and DNA ligase resynthesize the DNA strand. Mismatch repair depends on a number of activities in human cells, including MUTS alpha or MUTS beta, MUTL alpha, the exonuclease XO1 DNA polymerase, and the replication clamp PCNA, the clamp loader RFC, replication factor C, the single strand DNA binding protein, and the non-histone chromosomal protein. The first step in mismatch repair is recognition of the error, the method of strand discrimination. Which strand is the daughter strand with the remake mistake in mammalian cell is unknown. One model response is that 5' end of an Okazaki fragment and the polymerase machinery associated with the 3' end of the nascent strand may provide markers for strand discrimination and post-replication mismatch repair. XO1 in association with PCNA then progressively removes the portion of the strand in which the nick and the mismatch. HMGB1 plays a role in stimulating XO1 activity. Next, repair synthesis to replace the excised strand with new DNA is mediated by DNA polymerase and its associated factors. Finally, DNA ligase 1 seals the nick by forming the last phosphodiester bond. So, general steps of DNA mismatch repair: mismatch recognition, recruitment of related proteins, excision and replacement, and gap filling. Then mismatch recognition in prokaryotic MMR. The MUT-S protein complex detects the DNA lesion and combines with an ATP molecule and gets activated. In eukaryotic NMR, the MSH protein complex recognizes the non-matched bases in DNA strand and gets activated by an ATP molecule. Recruitment of related proteins in prokaryotic MMR, the activated ATP MUTS complex recruits MUTL, acts as a carrier protein. This MUTS and MUTL protein activates the MUTH complex. MUTS, MUTL, and MUTH form the ternary complex, which loads the DNA helicase 2 or UVRD enzyme onto the DNA lesion site. In eukaryotic MMR, the activated ATP MSH complex recruits PCNA and polymerase delta. Then ATP dependent conformation change occurs in the mobile clamp. The conformational changes recruit the binding of MUTLA complex. The PCNA proliferating cell nuclear antigens activates the MUTA complex and creates a nick in a daughter strand. Then excision and replacement. In prokaryotic MMR, exonuclease 1 is activated by the MUTSA protein, which removes the mispaired bases. Then RDA protein displaces the mismatched base. MUTSA and MUTLA terminate the exonuclease 1 activity. In eukaryotic MMR, the MUTLA complex promotes the digestion of the mismatched bases by the exonuclease activity 1 with the association of RPA, recognition, replication protein A. Then gap filling. In prokaryotic MMR, the DNA polymerase 3, through its 5' 3' activity, forms new bases. Single-stranded binding proteins SSB also bind to the new DNA strand and prevents its new DNA to become double-stranded. Finally, DNA ligase fills the gaps in the DNA strand. In eukaryotic NMR, The RFC, replication factor C enzymes, play an important role in the synthesis of new DNA bases. Replication factor C enzyme attaches at the 3' site and activates DNA polymerase activity to form new bases. Single-stranded binding proteins, or SSB, prevents the new DNA to become double-stranded. Finally, DNA ligase fills the gap in the DNA strand. Then excision repair. Nucleotide excision repair. The nucleotide excision repair pathway is used for repair of structural distortion. For example, bulges from thymine dimers induced by UV irradiation. In humans, nucleotide excision repair is carried out by 6 repair factors: RPA, XPA, XPC, like that. ERCC stands for excision repair cross complementing rod and repair deficiency. Bulky base damage, including pyrimidine dimers, can be removed directly without the help of DNA glycosylase. In this nucleotide excision repair pathway, the excising enzyme system recognizes the strand with the bulky damage and makes cuts on either side of the damage, removing an oligonucleotide with the damage. The key enzyme E. coli cells use in this process is called UVR-ABC endonuclease because it contains G polypeptides, the products of UVR-A, UVR-B, and UVR-C genes. This enzyme cuts the damaged DNA, producing an oligonucleotide that is 12 to 13 bases long, depending on whether the damage affects one nucleotide, alkylations, or two pyrimidine dimers. A more general term for the enzyme system that catalyzes nucleotide excision repair is excision nucleotide or excinucleases. Nucleotide excision repair takes two forms in eukaryotes. It can involve all lesions throughout the genome, global genome INER or GGNER. Bulky base damage, including pyrimidine dimers, can be removed directly without help from a DNA glycosylase. In this nucleotide excision repair pathway, the incising enzyme system recognizes a strand with bulky damage and makes cuts on either side of the damage, removing an oligonucleotide with the damage. The key enzyme E. coli cells use in this process is called UVR-ABC endonuclease because it contains 3 polypeptides, the products of UVR-A, UVR-B, and UVR-C genes. These enzyme cuts are damaged DNA, producing an oligonucleotide that is 12 to 13 bases long, depending on whether the damage affects one nucleotide or two. A more general term for the enzyme system that catalyzes nucleotide excision repair is excision nuclease or excinuclease. Nucleotide excision repair takes two forms in eukaryotes. It can involve all lesions throughout the genome, global genome NER or GGNER, or it can be confined to the transcribing strands in genetically active regions of the genome, transcription coupled NER or TCNER. Nucleotide excision repair typically handles bulky damages that distorts the DNA double helix. NER in E. coli begins when the damaged ATP is climbed by an endonuclease on either side of the lesion at sites 12 to 13 at NT apart. This allows the damaged DNA to be removed as part of the resulting 12 to 13 base oligonucleotide. DNA polymerase 1 fills the gap and DNA ligand seals the final nick. Eukaryotic NER follows two pathways. In GGNER, a complex composed of XPC and HHR23B initiates repair by binding to a lesion anywhere in the genome and causing a limited amount of DNA melting. This protein apparently recruits XPA and RPA. TF2H then joins the complex and two of its subunits, XPB and XPD, use their DNA helicase activities to expand the melted region. RPA binds to excinucleases and positions them for cleavage of the DNA strand on either side of the lesion. This releases damage on a fragment between 24 and 32 NT long. TCNER is very similar to GGNER except that RNA polymerase plays the role of XPC in damage sensing and initial DNA melting. In other kinds of NER, DNA polymerase D or F2 is the gap left by the removal of the damaged fragment and DNA ligase seals the DNA. Then base excision repair. In base excision repair or BER, a damaged base is recognized by an enzyme called DNA glycosylase, which distorts the DNA in such a way as to extrude the damaged base out of its association with its base paired partner, then breaks the glycosidic bond between the damaged base and its sugar. Next, base excision repair. So DNA is such a way that to extrude the damaged base out of its association with its base paired partner, and this leaves an apurinic or a pyrimidic site, AP site, which is a sugar without its purine or pyrimidine base. Once the AP site is created, it is recognized by an ATP endonuclease that cuts or nicks the DNA strand on the 5' side of the AP site. The endo in endonuclease means the enzyme cuts inside the DNA strand, not at the free end. Greek endo means within. Once the AP site is created, it is recognized by an AP endonuclease that cuts or nicks the DNA strand on the 59 side of the AP site. The endo in endonuclease means the enzyme cuts inside the DNA strand, not at a free end. Greek endo meaning within. Base excision repair typically acts on subtle base damage. This process begins with the DNA glycosylase, which extrudes a base in a damaged base pair, then clips out the damaged base, leaving an apurinic or a pyrimidinic site that attracts the DNA repair enzyme that removes the remaining deoxyribose phosphate and replaces it with a normal nucleotide. In bacteria, DNA polymerase 1 is the enzyme that fills in the missing nucleotide in BER. In eukaryotes, DNA polymerase B plays this role. However, this enzyme makes mistakes and has no proofreading activity. So APE1 carries out the necessary proofreading. Repair of 8 oxoguanine sites of DNA is a special case of BER. This can happen in two ways. Since OXO-G pairs with A, the A can be removed after DNA replication by a specialized adenine DNA glycosylase. However, if replication has not yet occurred, the OXO-G will still be paired with C, and the OXO-G can be removed by another DNA glycosylase and OXO-G repair enzyme. Then recombination repair. Recombination DNA repair is a biological reparative process in response to DNA damage caused by exogenous agents like UV light. This process is done generally generating functional DNA by breaking and rejoining DNA strands and repairing based on homologous unaltered DNA strand. The cell has repair mechanisms to correct DNA damage. For instance, a cell that is exposed to high energy radiation could cause the chromosome to break into two pieces. The cell repairs a broken chromosome by non-homologous end joining or by homologous recombination. The problem with the double-stranded breaks is the possibility of losing a large portion of the chromosome at the breaking point. To repair by a non-homologous repair mechanism, the broken ends of the chromosomes are joined back together. Since a portion of the chromosome might have been lost following breakage, the mere end joining of the chromosomal segment at the breaking site could likely lead to mutation due to lost nucleotides. This is somehow avoided when the repair strategy is by the more complex homologous recombination. In the homologous recombination repair process, the nucleotide sequence from the other pair of homologous chromosomes called homologue is an example of the template used to create a lost portion of the damaged chromosome. Homologous chromosomes are paired chromosomes in the nucleus. Chromosomes that have the same gene loci, gene sequence, centromere location, and chromosomal length, but may have different alleles are referred to as homologous. Humans in particular have 23 pairs of chromosomes. 22 of them are homologous autosomes. Half of them are paternal, whereas the other half maternal. These chromosomes pair up during meiosis, but not during mitosis. Recombination repair is a mechanism for generating a functional DNA molecule from two damaged molecules. It is an essential repair process for dividing cells because a replication fork may arrive at a damaged site, such as thymine dimer, before the excision repair system has eliminated damage. Then SOS response. The SOS response was discovered and named by Miroslav Redman in 1975. The SOS response is a global response to DNA damage in which the DNA cycle is arrested and DNA repair and mutagenesis is introduced. The system involves the REC-A protein, RED51 in eukaryotes, and REC-A protein stimulated by the stranded DNA is involved in inactivation of repressor LEX-A of SOS response genes, thereby inducing the response. It is an error-prone repair system that contributes significantly to DNA change observed in a wide range of species. Damaged DNA inactive. REC-A. SOS repair refers to the DNA repair system which uses REC-A regulatory protein to inhibit the repressor's activity and activate the SOS inducer gene to recover the DNA damage. It stands for Save Our Soul. Save Our Soul SOS system remains repressed until the conversion of REC-A protein into REC-A protease. It does not repair the DNA damage completely but provides tolerability to the affected organism. SOS system only activates in case of excessive cell damage leading to single-strand breakage at the replication fork. This DNA damage activates the REC-A regulatory protein that links with the single-stranded DNA through a cellular energy ATP. Mechanism of SOS repair. The mechanism of SOS repair is a complex cellular process mediated by the organism itself. It includes the following steps. In case of excessive DNA damage, stress conditions, a cell responds by activating signal or REC-A protein. It floats in the vicinity of the cell in search of any damage in the DNA. REC-A protein specifically binds to the single-stranded DNA. In binding with the single-stranded DNA fragments, REC-A forms a filament-like structure around the DNA. Then a hex-A repressor comes in contact with the nucleoprotein filament assembled by the REC-A protein. When REC-A interacts with the repressor protein, it converts to REC-A protease. The formation of REC-A protease causes autolytic proteolysis of LEX-A repressor protein. Thus, a LEX-A protein could not bind with the SOS operator. Inactivation of LEX-A protein activates the inducer proteins that repair the DNA damage but alters the DNA sequence. After DNA repair, the REC-A protein loses its efficiency to cause proteolysis and the LEX-A protein will again bind to the SOS operator or switch off the SOS system.